myself first. So this is our very first Microgrant meetup, and I'm Ryan Kelly. I'm the director of the eDNA Collaborative, and I'm sitting here in Seattle, Washington, which is at the University of Washington, which is the upper left-hand corner of the continental United States. And we started about 18 months ago with this mission to move environmental DNA methods out of the lab and into routine practice around the world. And we thought, well, how could we do that? What are strategic ways that we can make eDNA work better for more people and make it useful in practice? And so we've explored a few different ways of doing this, and these are listed on the screen here. We engage in core research. Part of that is, how does eDNA work? It just fundamental science, and then can we make it better, cheaper, faster for people, and so forth. We've had we've been able to offer a few seed grants to people as sort of demonstration projects. We have visiting scholars. We've had 6 of these so far who have come and sat with us in a two-way exchange of information, and we've learned a ton, and they've learned a ton, and that's been fabulous for creating community and creating a network of people that can work with each other and move the science of eDNA forward. But microgrants really has become central to what we do, and you are all on this call because you've been funded at some low level. We thought, can we support a lot of people with a little bit of money, and that way reduce barriers to entry into a field that can otherwise be expensive to get into. And so the thought there is, by reducing barriers to entry, making eDNA more accessible for more people, we create a bottom-up demand for eDNA methods around the world. Now, that has been great. We've got over 100 microgrants awarded to people in over 40 countries, obviously all around the world, and that's fabulous. And then the next step of that starts today. What we'd like to do is stitch you together into a network of people who we know each of you at some level. We know your name. You do not know each other. And so that's the point of today. And the succeeding 2 versions of this meetup, we broke it up so that people could be participating no matter what time zone you're in. So we're offering this same adventure 3 times. You are more than welcome to join more than one of them, but that's why we have this in different sessions, because in order to facilitate participation in time zones around the world. I'll also mention we have closed captioning or available for this meetup, and there are others. So if you are in the Zoom app, you should be able to see a button that says show captions. And we realize, of course, that English is not everyone's first language. It's actually most people's not their first language. So if it makes things easier, that caption option is available to you, and you should have that button on Zoom. And in any case, that's the goal, and the process, and the point today is to start making these connections across continents, across people, across research areas, so that you can reach out to each other and have potential collaborators and potential friends to call upon for help when you need it. Let the other members of our team introduce themselves here, and then we'll be off and running.
Thanks, Ryan. Hi, everybody. My name is Kara Sucher, and I know I've been in email contact with all of you at least once. So it's really great to see you, and I'm very much looking forward to hearing about all of your work. I joined the collaborative about 18 months ago. My past experience is when I was doing science, I was an engineer, and then I was a geochemist for a little while. I did paleoclimate research, and then I spent the majority of my career working for the U.S. government and in support of scientific research down in Antarctica. So my background is, the majority of my background is sort of logistics and coordinations and operational aspects of science. That's me. Eileen?
Thanks, Kara. Hi, everybody. My name is Eileen Allen. I'm the chief scientist here at the collaborative. I'm super happy to meet you all. I've maybe emailed with some of you, but not quite all of you. So really excited to hear about all of your work. And a little bit about my background. I actually started doing eDNA just over 10 years ago as undergraduate research. And then I got my PhD, and my whole dissertation was around using environmental DNA, which was pretty new still at the time. And I mostly work in marine and but really in the collaborative, it's been really interesting to see, water, air, soil, everything. And so really excited to learn from all of you. So thank you for joining us, and thank you for participating. I'm going to just do the first introduction for the first presenter. And then, as Kate mentioned, if everyone can introduce themselves after that, the slide order is on the Google Doc. And we're going to give everybody 3 minutes. If you're getting close, I will just give you a gentle reminder either in the chat, or I can just unmute and say, 30 seconds left. So first up, we have Camilla McLaughlin.
Hi, everybody. Can you hear me okay? Yeah. Okay. Okay, so my study is the whole of my PhD dissertation as well, which is also still pretty new in Mexico. What I'm showing here in my slide is sort of the latest that I've been working on. It's an integrative approach to compare biodiversity of cryptobenthic and conspicuous fish in the Gulf of California. The Gulf of California is a marine biodiversity hotspot known for having 3 distinct regions. So our objective was to compare what scuba divers see on their visual surveys against fish captures, which is only the most effective way of monitoring cryptobenthic fish, and just looking into what eDNA can do for either of these groups. So we did a standard approach, sort of like the least cost possible scenario, 1 liter taken on 30 different places in the Gulf. We did the teleo barcode with the standard 2 PCR, 2 adapters. I used the Anacapa pipeline to get amplicon sequence variants, and did community ecology analysis in R. So what we found, if you see my tiny plots, is that conspicuous fish have higher richness in the central Gulf, but that pattern is not the same with cryptobenthic fish. We found sort of similar richness between regions. And then the contribution of each method to fish diversity, it's on this, pie chart. So we have 60 cryptobenthic fish. Most of them were found by captures, and there is not a lot of contribution from either eDNA or from visual surveys. But for conspicuous fish, you can see it's a more shared effort between eDNA and surveys. They're sort of showing us the same amount of species each, and not a lot of them are found in captures. So eDNA is not really a great complement for cryptobenthic fish if you're considering this least cost scenario, where we just sent 1 scuba diver down the water and took a liter of water, sort of like 1 or 2 meters far away from the bottom. So that could be a consideration. Nevertheless, we found both groups of fish show isolation by distance, and we found with less agreements, but also this distinction between north and central regions, considering beta diversity. In the first part of my PhD, I also analyzed the 18S barcode and I found a similar pattern for invertebrates, algae, fungi, and other eukaryotes. So my future investigation is going to be what is driving these patterns of diversity that are shared among all these different groups of animals. Thank you. Next slide, or next person.
Hi, thanks a lot. Hi there, my name's Clio Hall. I'm a PhD student at the University of Helsinki in Finland, but hello currently from the ICES conference, which I saw people were listed there, where I were just presenting my results today. There's a slightly snapshot version of them, but basically my research looks at climate change happening in the Baltic Sea, particularly freshening and warming that's going on there. And I look at how plankton food webs respond to this kind of climate change. using both eDNA methods and trying to compare with microscopy. So I'm using actually kind of an experimental setup. I mainly work with mesocosms, which is what you can see from that little picture there. It's my different tanks have different treatments in them for kind of warming and different salinities. And so then basically my last experiment ran for approximately 5 weeks. And I've been looking at trying to assess how the plankton community changes with these treatments with both microscopy data as well as DNA data taken throughout the experiment. So yeah, then I assigned I was using 18S and CO1 markers, and I assigned this taxonomy using these Midori 2 and fancy new PR2 databases. And basically, just to give you a flavor of the results that we found, basically, I was using these kind of like newly developed analyses, which are actually meant to be for gut microbiome analysis. And they try to kind of like minimize some of the problems that surround DNA data, like the biases around certain taxa, stuff like that. But I mean, you can have a look at the papers if you're interested in that, especially if you're working with experimental kind of data. But basically what we found was that the effect of salinity was not so much found in both microscopy and the metabarcoding data, but that there were very complementary results found for the effects of warming, which mainly seem to affect these kind of smaller processed communities were being affected by warming, leading to kind of the smaller size zooplankton, which is not so good news for fish, and also a lot of these nasty bloom forming freshwater algae happening is the kind of future scenario for the Baltic Sea. So I'm interested in anyone having similar kind of experimental data, or just here to hear what everyone else is doing, basically. Thanks a lot. Next slide.
And I've seen Jessica coming in and out of the Zoom. So maybe she's having connection issues. We'll check back with her at the end of the meeting, unless Jessica, you're on. All right, next slide.
Hi there, I'm Josie Pegg from South Africa. I'm based down in Kruger National Park, and yeah, so the work that I'm hoping to achieve is looking at the distribution of a fish pathogen in South African waters. This is a pathogen that causes a ulcerative syndrome. So it is sort of quite damaging for sort of food security, and it's a global problem. It originated from sort of Asia, but it's problematic in Australia. The reason I actually got interested in it, I have colleagues, I used to live in the UK, and I have colleagues in the UK who are sort of very interested in developing sort of simple ways of detecting it in water to sort of forecast its arrival in Europe. And they asked me for samples because we do have it here. But then when I started looking into it, I realized that there was no information about the extent of this pathogen. We certainly have it north of South Africa, so in Zimbabwe, Zambia, Botswana sort of areas. If you look on the map here, sort of above Swaziland, you can see 2 river catchments.
Oh no, we lost audio, I think, there. It seems, Josie, you said we're frozen. Okay, let's give her just another second there.
I think she dropped off, so maybe her internet went out. So let's move to the next one, and then we'll kind of come back at the end for
Great.
Josie.
We'll circle back to Josie.
Hi, thank you for the time. Hope you hear my voice. I'm Harus Saputra from Indonesia. This time I would like to give a presentation about one of our projects related to eDNA. It is about study of aquatic organism diversity in Batanghari River using eDNA. For this project, we have several people involved in this project, 5 people from National Research and Innovation Agency, BRIN, Indonesia, and also we have people from Sriwijaya University. Sorry, yeah. Okay, the background of this study, in addition to climate change issue, the anthropogenic activities along the river result in a decline in the aquatic organism diversity, including fish. We focus on the Batanghari River because it is one of the longest river in Indonesia, located in Sumatra Island, the Jambi province of Indonesia. This river is also polluted by mercury-containing illegal gold mining waste. So this factor obviously affect the aquatic organism diversity in this river. So in this study, we try to evaluate in the river using eDNA approach. So the method we use here, the water sample we collect from 15 sites through 5 major watersheds. Start from the upstream to the downstream of the river. Use analysis, and then after that we conduct a next generation sequence analysis. Then what we found from this study is that its major watersheds show significant variation in aquatic organism diversity. As you can see on the graph on the right side, you can see that each the watershed or the branch of the river has a different profile of organism diversity. Of course, this is still a preliminary result from our study, a lot of work to do, but we expect to publish the data maybe next year, maybe at the beginning of the next year. So for future collaboration, and we realized that we are still struggling to analyze our eDNA data because we don't have an expert for this analysis, more for bioinformatician. So perhaps from this forum, from this meeting, maybe we have, there is someone who can help us to analyze the data or to visualize our data for publication purpose, for example, like that. And then another thing is that in the future we want to try to apply this the same approach to study, to monitor the current status of the organism in the lake, not only in the river, So maybe if you are interested, maybe we can collaborate together in the future. We can create a proposal to study, because in Indonesia we have some lake, big lakes. Those lakes are facing the same issue with the river. We concerned about that, about the diversity of the organism. So yeah. Maybe some of you interested, and we can talk later, and then we can conduct the similar research in the lake. Yes, I think that's all from me. Thank you. And next slide.
Hello, everyone. I am Cornelia Tuhrinova, and I am here with Andrej Vargovčić because we are in a field now. We are collecting eDNA samples, and we are from same lab, so we are using one computer. I'm sorry for that. So my name is Cornelia Tuhrinova, and I am PhD student at Comenius University in Bratislava. The main aim of my study is assessment of the use of environmental DNA as an alternative to conventional methods of monitoring the biodiversity of alpine lakes. Tatra Mountains represent a very unique small area. Tatra lakes have very simple communities sensitive to changing environment, and they are also served as a refuge in Ice Age. So for that they are this is a good area for studying. And we have eDNA samples from year 2019 to 2022, but we will also, after this week, we will also have samples from 2023, and we are collecting 3 types of samples: eDNA from sediment, eDNA from water, and a bulk. After we extracted DNA with a question blood and tissue kit, and we are using one barcode, and I don't have many results now because I'm just starting to analyze everything, but we want to compare eDNA from sediments and eDNA from water, and then maybe DNA from bulk. And we also have some results, and in some lakes we capture of terrestrial fauna around lakes. Next slide.
Hi everybody, I'm Luciano Pastorelli, currently undergraduate student from Peru, and my studies called Fish and Benthic Invertebrate Diversity on the coast of Central Peru. As a background, the study site is located within the Humboldt Current System, one of the most productive currents worldwide, and so it's very important to know how biodiversity behaves within the system and how eDNA metabarcoding may perform as a tool for monitoring biodiversity. Several phases of biodiversity will be evaluated, such as taxonomic, functional, phylogenetic, and also trying to understand the spatial patterns obtained with these methods. And it is located within a large marine infrastructure, so it is we are doing waters and tests for fish, and we are currently developing a bioinformatic pipeline. Apart from this, DNA may be used as an alternative or complementary tool for monitoring biodiversity associated with this infrastructure. Currently, results are not available, are processing right now to join a venture service in Boulder, Colorado. And apart from this study, well, I also collaborating with one project that tries to understand how organic matter might affect biodiversity, and also another study that wants to understand how linear currents, a linear phenomenon, might be affecting biodiversity, fish diversity. So that's helping me on knowing how eDNA works, but I have never processed samples in like lab for eDNA metabarcoding. So I'm willing to perform. I'm searching for training courses on this, and also to support further studies to conduct other ones here in Peru. And next slide, please. Thank you.
Well, hello everybody. So my name is Mark Abramov. I'm a student, I'm a PhD candidate at Carleton University in Ottawa, Ontario, Canada. My study focuses on mosquito-borne diseases and finding ways to improve surveillance and monitoring. Specifically, I'm studying California serial viruses. So they're not really known worldwide, but they're encephalitic viruses that are prevalent in Canada. And they are amplified via small and large mammals. So basically right now I'm in the field, I'm collecting mosquitoes all day, which is a lot of fun. I'm also collecting mammal footprints using these cool tunnels that have like ink pads in them. And basically right now what we know is that we have several mosquito pools that are positive for these viruses at my sampling sites. And we also know that the hosts, those amplification hosts, are present at our sites as well. We're trying to figure out a way with my supervisors to collect eDNA from those ink pads to see if we could potentially capture virus presence within these ink pads. I know it's been done for other viruses, but it's literature is a little bit scarce at the moment for that. So I would love to talk with any of you that might have any idea on how to just swab and run some eDNA on analyses on these papers to figure out if we could capture hotspots for these diseases at my sampling sites. So yeah, there's a lot of other things, but we'll keep it at that. Just for time's sake. Next slide.
Hello, my name is Ondrej Vargocik, and as Cornelia has already introduced the topic of that we are dealing with in our team. Here you can see also the location of our field where we are right now in the High Tatra Mountains of the Carpathian chain. So maybe one thing that wasn't said for now is that we are mostly focused on invertebrates present in these lakes. And we also had some experiments with the ethanol, the storage ethanol that is used for the benthic kick samples, the bulk samples. And maybe that's one part of the results that I can already present. We made a small experiment there about how quickly should we process the samples, if there is any advantage of leaving the samples to stand and stand in the storage ethanol for longer. And I was a bit surprised that several studies have doomed the method, especially for terrestrial and marine environments, that the fixative is not very useful. That it cannot even distinguish between lake types or habitat types. But here we can see the two outgroup lakes, which are the in the bottom right graph there, the low altitude lakes, and also other gradient other signs of gradient there in the graph, which I'm not diving into too much now. Yeah, we have also identified quite some match with previous studies, like the composition of fauna there. And for the future, we are looking forward to apply this in the Water Framework Directive monitoring, which is already done in our group by another colleague. We also try to apply these methods for some pollinator research. Yeah, I'm I'm looking forward into like doing these analyses and finding out as much as we can about the invertebrates and other kingdoms of life. So thank you, and next slide.
Hi everyone, my name is Shannon Dickey, and I am a PhD student at Rutgers University in New Jersey. As a marine ecologist, I'm really passionate about conserving biodiversity, and so today I will be talking about my work on understanding the impact of tides on fish environmental DNA in a salt marsh. And so I became interested in salt marshes due to many reasons, such as their ability to have very high biological productivity, acting as blue carbon sinks, and protecting the coastlines from storms. So what not to like, am I right? However, despite their importance, globally marshes have experienced a 50% decline in size, either being degraded or lost entirely, most part due to climate change. So in response to this crisis, nature-based solutions are an emerging response to mitigate the impacts of climate change. And despite their known effectiveness, there is a lot of limited research that examines the long-term impacts of nature-based solutions on biodiversity, especially in tidally influenced systems. And as a result, eDNA metabarcoding can provide an additional way to assess the biodiversity benefits and nature-based solutions in marshes. And that is what the bulk of my research focuses on. And so what we set out to do in this project was to answer three main questions. In image A, we wanted to estimate the temporal scale of an eDNA signature with regards to tides, so that we knew when would be the best time to sample to get a complete picture of the species composition then. In image B, we wanted to understand the diversity of fish in a marsh pool, and this allowed us to compare different primer sets and see which would be best for our system. And then in image C, we wanted to see if using a traditional method like a plankton net for eDNA purposes could gather more information about the fish diversity than a traditional method could do alone. And so what we did was sampled our eDNA net alongside a traditional plankton net sampling group, and then we came together and we compared those results. Now, to answer these questions, we had very similar methods where we collected a 1 liter aquatic samples from the Jacques Cousteau National Estuarine Research Reserve here in New Jersey. And we did this at 4 tidal cycles, which were at the low, the incoming, the outgoing, as well as our low tide. And so after we collected the samples, we transported them back to the eDNA lab at Rutgers, where we extracted the samples, amplified them, and hopefully soon we'll begin to identify the sequences bioinformatically, which is very helpful because I took one of the courses with this grant. So I'm excited to get that going. And so everything's going well so far in the lab. I have compared 2 primer sets, which you can see on the side here. I've got the MyFishU by Mia et al. 2015 and the Eco Primers by Rios 2011. And based on these very preliminary results, I am excited to stick with the MyFishU primer set and to continue down that path. And so overall, this project was a collaborative effort between the Rutgers University eDNA Lab, the Rutgers University Marine Field Station, as well as the Jacques Cousteau National Estuarine Research Reserve. And with their help, I'm working on constructing a very user-friendly protocol to answer key questions about fish diversity in salt marshes. And to keep with that same theme, my next goal is to take our marsh pool sampling protocol and apply it to assess rare bird species in New Jersey, like the black rail and salt marsh sparrows, so that we can inform more adaptive management plans to conserve marsh habitat. Also in my future, I hope to continue my efforts in measuring the co-benefits of nature-based solutions, specifically biodiversity in coastal ecosystems. Thanks, guys. Next slide.
Hi, everyone. Can you hear me? Yeah, great. Thanks. Thanks for organizing this, and thanks also for funding our research. So my name is Stefania, and I'm associated with the National Museum of Natural History in Sofia, Bulgaria. And I have been working with DNA metabarcoding diet analysis for the last 10 years. And I'm usually more focused on large herbivores such as reindeer and bison. But I have this small project that is very close to my heart, focusing on the mouse-tailed dormouse, or Ruache's dormouse, which is a really cute small mammal with a very narrow distribution within Europe. It's actually one of the rarest dormouse species in Europe. And there is only single population in Bulgaria, and it's actually extremely restricted range for the species. And with colleagues from the Natural History Museum, we set up research plan to establish an action management plan because nothing is known about the species, and this prevents us for actually taking any conservation measures. The species is listed as vulnerable by the UICN. And as part of this effort, we have set up this regular monthly monitoring since 2019, where we actually capture, measure, and tag all the individuals. And we also collect fecal samples for diet analysis, which was very generously supported by the eDNA collaborative. So we have been targeting plants and arthropods in the diet. We have extracted more than 300 fecal samples. PCRs were done, and now we are awaiting for high-throughput sequencing. And in terms of, I guess this is not the right audience for this, but we really do need to collaborate with people in expertise in radio and GPS tracking in order to figure out where actually species forage and link potentially this information with diet composition. So I think that's basically everything from me. So thanks again, and next slide.
Hi, my name is Josh Gunguchlu, and it's been really great to hear about all your projects so far. I'm based at the University of Hong Kong, where I'm entering my final year of my PhD, and I'm using eDNA to study aspects of secondary succession in Hong Kong tropical forests. So Hong Kong is a really interesting place in terms of its forest ecology, because basically all of the forest in the area was completely destroyed during the 20th century and is now regenerating largely spontaneously. But there's also some parts of Hong Kong where government agencies and NGOs have been trying to restore forest. And my PhD research really revolves around asking questions about the consequences of restoration for things like biodiversity, as well as ecosystem service provision. So prior to conducting my project using eDNA, I actually explored how plant community composition changes during secondary succession. But now we really want to turn our attention to looking at things below ground. So we've been using 16S and 18S to characterize the soil biome, and focusing largely on arbuscular mycorrhiza and also just the soil microbiome, basically. And we've been doing this across a set of 24 sites, which are split between forests that have been passively regenerating and forests that have been replanted in a very specific and intentional way. And these are further subdivided into different types of reforestation treatment. So we're hoping that the data that we'll get from this project will be able to tell us things about the value of sponsoring things like forest replantings, and whether there is actually a benefit to just abandoning land and allowing it to restore on its own. But one final aspect of this project that I think is really exciting is that we're trying to look at ecosystem service provision alongside this community structure of the soil biome. So we're also measuring the carbon content of soil and the carbon respiration out in and out of the soil. And at the moment, I have no results because the PCR is ongoing. So I just finished one a little bit earlier today, and it's all coming along really well. We're hoping to send off our PCR product for sequencing at the end of the month. So watch this space for more. In terms of collaboration, we'd be really interested to reach out to people who are conducting similar kinds of investigations, because I think it's really interesting for us to think about how we can compare different systems and see whether there are commonalities as the result of rewilding and forest restoration. So that's been my research, and thank you so much for listening. Next slide.
Hi everyone, can you hear me okay? Yeah, cool. My name is Aideen O'Mahony. I'm usually based in Scotland at the University of St Andrews with the Sea Mammal Research Unit there. But I'm currently in Gitga'at First Nation territory, carrying out my final set of fieldwork for my own PhD. We're working collaboratively with the Git-Gab First Nation and a not-for-profit organisation called BC Wales. And I'm lucky enough in that the PhD gets to build on about 20 plus years of humpback whale and fin whale data, social and cultural data using photo ID methods and tracking these whales through the fjord systems through time. And kind of the missing puzzle piece for a lot of the work that's ongoing here is the underlying genetic relatedness patterns of this population of humpback whales. And so the first part of the PhD that I thought was relevant today is using a small drone to blow sample humpback whales. And the photograph you're seeing is a humpback and the drone flying through those droplets of breath. And I'll be shotgun sequencing those blow samples to get ideally some SNP genotypes for these whales and map this genetic relatedness pattern of these pairs of whales across and compare that across their social network that we've previously analyzed together with colleagues here. These same humpback whales, as the feeding season for these whales progresses, they move deeper into the fjord system that's circled in red. They're moving kind of inland in many ways into these fjords. And then in September and October time, they congregate in very small areas, sometimes 50, 60 humpbacks at one time, might be in a very small space of about 4-5 square kilometers. And the question that we have is, what is driving these congregation sites or these hotspot sites for these whales? It doesn't seem to be as simple as just following their prey into the fjord system. So that's where the seawater eDNA is coming in. We'll have the blow sampled relatedness pattern for this congregation site or these hotspots, but we're then using eDNA sampling across 2 hotspot areas into not hotspot or not spot, I guess, areas to look at the community composition, the underlying biodiversity of these spaces to try to get at some of these drivers for these humpback whales. Our sense is that it's probably some kind of a cultural element, but we'd like to rule out any kind of prey following to begin with. So yeah, so that's kind of 2 parts of the PhD. When I was thinking about future collaborations, I recognize now that they're very, very specific. So 2 projects that I'd like to see happen or somehow be involved in is a custom designed SNP panel for this population of humpback whales, because we're shotgun sequencing the blow samples, because I just in the PhD right now don't have the time or the money to custom design a SNP panel for them. And the second one I thought of was using this shotgun, applying more of a metagenomics approach to the data that we'll get from these blow samples, because I'll only be working with the humpback DNA, but we'll have everything else, the data at hand. And the very last thing I'll say is that a more general interest of mine in other projects in my PhD that I haven't touched on today is looking at methods of decolonizing ecology and evolution research. So how can we as researchers adopt more ethical approaches to our research, particularly when we are working collaboratively with Indigenous peoples and in Indigenous territory. And so I'm always on the lookout to connect with other researchers or people who work more in a field system to look at how we can better our research methods in a decolonial way. So yeah, I'll leave it at that. I'm not sure if we're looping back or if there's a next slide. Thanks.
All right, and we'll head back to check out Josie's slide. Josie, are you back online? And everyone stay tuned.
I am, yes.
Go through slides
Yes,
quickly. Whoops, here we go.
Sorry about that.
No problem.
We have rolling blackouts at the moment, but sometimes we also have surprise blackouts, so you never really know. Yeah, anyway, so I think I'd got to the point where I was sort of I'd introduced the background and yeah, the fact we once we started looking, it became obvious that we really didn't know where this disease was, even though we knew it was present. So yeah, so that's sort of the state of affairs. It is a aquatic, it's a fungal pathogen. We have sequenced it, so we know what we're looking for, but yeah, we haven't got any results as yet. September is sort of the best sampling season here. It's very, very hot. We have very hot, wet summers. So we're just sort of coming into summer now. So it's a really good time to go out sampling. So no results yet, but hopefully some soon. But yeah, the sort of future collaborations. Firstly, I'm interested in anybody that's working on sort of aquatic, particularly fish, but any sort of aquatic disease detection using eDNA. I'm actually an ecologist. I have done some sort of disease ecology, but I haven't done sort of this is only my second sort of foray into eDNA. So yeah, I'm interested in anybody else working in that field. And then sort of more generally, we South Africa is a utterly beautiful country. We have amazing rivers. We have reasonably good sort of genetic sort of study capacities, but eDNA is really, really sort of low in terms of our sort of progress here. There's really only a couple of really good sort of eDNA studies. So if anybody is particularly sort of freshwater, the marine sector is sort of growing, but certainly in the freshwater sphere, if anybody is interested in anything from sort of general detection of fishes or any sort of other sort of related aspects, yeah, lots of opportunities to do work here, and I would love to welcome you. I have a lovely field station in the middle of Kruger National Park, so yeah, and huge thanks to the to yourselves, eDNA Collaborative. I know there are any microgrants, but $1,000 goes a huge distance in South Africa. So thank you very much for your support of my work. That's it.
Great, and we'll go back to Jessica. And Jessica, apologies for jumping past your slide quickly last time.
Thank you so much. I'm sorry I couldn't do this at the right time. I had an audio issue here, so that's why I'm using my cell phone to do the presentation, but let's do it. So I'm Jessica, I'm from Brazil. I'm a PhD student at the University of São Paulo, and the title of my work is Unraveling the Role of Plant Growth Promoting Bacteria in Recruiting Microbiome from Maize Rhizosphere. So my work is a little bit different from what we have seen so far this afternoon because I'm working in the plant agriculture part using eDNA. So we have plant growth promoting bacteria, specifically two strains that I'm studying my PhD, which are Bacillus thuringiensis, BT. It's a strain isolated from Amazon, from a native tree in Brazil in the Amazon region. And we also have this number 2 strain, which is Azospirillum brasilense, the strain ABV5, which has been used commercially in Brazil, as this strain is a natural nitrogen fixer. So it substitutes mineral fertilizers in agriculture. But the problem is that the lack of holistic comprehension about PGPB plant native soil microbiota can lead to inconsistency of results in field conditions that we have seen here in Brazil, since we have a continental huge country and we have a variety of conditions. So we wanted to use eDNA, especially from plant-associated bulk soil and rhizosphere, to understand how this plant growth promoting bacteria recruit microbes from native soils to help plant grow. So the plant microbiomes are organized as microbiohubs with keystone species and helpers that can influence plant metabolism and physiology. And based on these concepts, we are trying to develop a synthetic bacterial community, including these two strains, these two PGPBs, as keystones for plant growth promotion. So on the left side of my slide, I have two traits evaluated in maize that were significantly increased when we applied a wild type bacteria that has a quorum quenching activity, which interferes in quorum sensing from other bacterial species that can cause damage to plants. So by applying our bacteria, our strain BT RZ2-MS9, we could see a significant increase in shoot and root dry matter opposed to what we have seen in the mutant strain, which does not have the corn quenching mechanism. So this strain is a promising biocontrol agent because of this trait, and we have proved this in greenhouse experiments. In the central picture here, I have metaomics integration data that we use to understand the role of the strain ABV5 to recruit both function and bacterial taxa from the native soil microbiome and how this helped plant growth. And on the right part here, on the last two plots, we have the data that we collected based on our greenhouse experiment, where we applied the synthetic bacterial community that we have developed based on metaomics data integrated. And we have seen that this income can increase plant growth rates in greenhouse conditions. So basically, the methods that I used were metataxonomics of 16S rRNA gene, metagenomics, and this data integration, also plant phenomics, and qPCR to track specific strains in the microbiome. And the collaboration needs that I see in my work is to have people helping me in the network analysis of microbiome communities. I have some interesting data and I want to improve these networks. So thank you so much for this opportunity, and we're open for questions.
Great, and I'm going to stop share so we can get to the question and discussion and everyone can see everyone else.
I'm like exhausted already because I'm like typing as fast as I can. And I know Eiley feels the same way. I was just trying to get to the question about suggestion of working with indigenous groups in ways that are sort of productive and meaningful. And the link that I was looking for was with John Hagen has been doing some work with the Northwest Indian Fisheries Commission in the United States, which is a collection of tribes, and he's an employee of the tribes, working closely with them, and so funded to work to be the eDNA person for managing salmon populations here in Washington. So thank you, Elif. That's the Elif has just posted in the chat the link to the organization, the Northwest Indian Fisheries Commission. Our contact there is John Hagen, and we can connect you with him if you remind us. We can do that. I'm realizing the challenge here is processing power. We're like doing 10 things at once and none of them well. So this is meant to be the part of this meeting. We've now just heard from people doing very diverse work, very interesting work. There, it's Hagan, John Hagen. Yeah, thanks. So many, I've got like 3 documents open all at once and I need to focus on talking. So the point here is to be able to have a discussion among people and have you be able to talk to each other directly. And I realize people can be a little bit shy on the camera. And so I wanted to start out with a suggested question that maybe for many people, it seems like there's a limiting factor in bioinformatics and analysis, and in particular, like annotating taxa, so that you can put the names of species onto the names to the sequences that you have. And I wonder, a question for the group, is that your limiting factor? And if so, do you have suggestions for how have you succeeded in overcoming that, or have you? And if not, we can help. Please, Stefania.
Yeah, it's actually quite good point, and more and more confronted to this problem working in Bulgaria, because it's such a highly diverse place, and it's a bit like this a vicious circle of where to start with. For instance, in my case, with the dormouse diet, So we cannot really start building a reference database because we don't know how diverse and which are the main target organisms these little guys predate on. So it's a bit like, yeah, let's start with something. We would expect that we wouldn't get a really great taxonomic resolution, probably family for the plants and orders for the arthropods. But maybe just delineate some main or common groups and then probably start referencing those. But it's a huge effort every time, and you need the taxonomic experts. We don't have experts for all groups. So, yeah.
Yeah, Diego, do you want to chime in, and then I'll add something at the end?
Yes, thanks for the question, and I apologize for not being able to turn on the camera. I'm on precarious means. I'm using a mobile device right now. So actually, I appreciate the topic because it gives importance to the kind of investment in time that I have decided for my career, because I myself saw this bottleneck in genetic study for especially applied to ecology, which is bioinformatics. It is good that right now it has been also like acknowledged to a broader audience, and so most of the time, answering the like the question what to do with the lack of expertise. Most of the time, when it comes to eDNA, it usually entails metabarcoding, which means it is about annotation, taxonomy. And so, on one hand, the bottleneck lies in the availability of good references, which has already been addressed before by Stefania, if I'm not mistaken. And yeah, I mean, if references are not good, one could start from scratch and with the barcoding together with morphological taxonomy, especially for the morphological taxonomy, very time consuming. Another solution, of course, might be to invest more time in the actual training. So gathering as a way to gather more expertise, and which can help also in the building of reference databases, because some pipelines like OB2s can help build customized reference database. This is also a very hot topic as of now that is also acknowledged, but it goes never out of fashion. The importance of having reference databases that would address, for example, geographical variants, local haplotypes that might not be present at in the more widespread databases. Yeah, I would say.
Okay, I know, Ayla, you said you had a comment, but maybe do you want to let Cairo go first?
Yeah, of course, please.
Yes, thank you. As I mentioned in my slides, yes, we had a problem. We are struggling to analyze our eDNA because we don't have an expert to analyze the data. Actually, we try to analyze our data. We use a web platform. It's called Galaxy Bioinformatic Analysis. We try to use that one, and then because we try our knowledge to analyze. We limited knowledge to analyze the data, and then we have a problem with the database. We only it's difficult to get databases to annotate our sequence, right? And then at the time we use a database from NCBI. And then we try to annotate. The problem was that sometimes because we are working on the freshwater environment, and we try to detect a fish from the freshwater environment and from the river, but some of them we detect a fish from from the marine. So we don't know what the problem. Maybe we have a problem when we how to say, when we assembly the sequence, maybe. So that's why maybe yeah. Thanks to Kelly to suggest me to contact Dita. I actually we know her, but we haven't tried contact. Maybe that's a good idea to contact her for this problem. Yes, I think that's. Thank you.
Awesome. And if anybody else has anything to add to this discussion, I was just going to add that oh, a hand just went up. Cleo, you go first.
Yeah, sorry, it was just a quick comment that I just was gonna chime in that I kind of like found converse results, whereas I actually had a really good species, like to species level and genus at least level identification of things that I had in my samples with metabarcoding that I'd also found with microscopy, sometimes even a bit better than I did with my microscopy. But what really like the problem that I was having is like how the DNA data responds to kind of like the ecological question that I'm trying to answer, specifically like with my research kind of using eDNA to understand the response of kind of climate change in these species that you're looking at. It just doesn't seem to necessarily be kind of, you know, it's a question of these, maybe the abundance stuff that everyone's kind of talking about, but it's just, my problem is basically with the nature of the response is not kind of a linear response with microscopy. But yeah, I just want to chime in for that.
Oh yeah, we could talk about that all day long.
Yeah, we could. That could be a whole nother hour. It's actually a great segue to what I was going to say about databases was also just, you know, I heard from a lot of the themes of everyone's short 3-minute talks is a lot of you are in high biodiversity places and the databases are limited. And it's the databases, but also the markers of choice. And so I thought it was interesting to see people who mentioned exactly which markers they were using. And so maybe trying to kind of connect with each other as well. A lot of people are using CO1. I assume those are the Luray primers. And that kind of just goes to exactly what you were just mentioning, Cleo, which is on the kind of research side of the collaborative, we have been doing a lot of work with that question of how different primers, the combination of the primer and the species of interest is really important to answer to look at those patterns and look at things like abundance. And so primers with degeneracies like the CO1 primer are really beneficial because they give you a really, really wide range of taxa across the tree of life. But it makes those detections a lot more stochastic and random of what you pick up where, because the reality is it's not one primer set, it's many, many primer sets in one tube. And so the being careful about the primers, what species you actually care about, the primers you're using, and of course you're limited by the databases. Maybe you're using those primers from CO1 because for your area, that's where the database is most populated. So just, you know, when we think about here, what is most useful for people to learn, it really is a powerful tool, but there's a lot of decisions along the way that you make that could impact your ultimate question, Cleo, which you were just mentioning, which is how does this relate to the ecological question of interest? So just wanted to add that. If anybody else has a new topic, we also have the questions in the chat. Please feel free to use the Google Doc for that as well. It's really fun to see all the connections being made in the chat. So, you know, keep that going as well.
Can I add to your to that point too about the markers and being able to distinguish what's sort of random stochastic amplification and detection of species versus systematic responses to an ecological question of interest. And this speaks to Khairul's question also, right? That why do you see unpredictable patterns in the DNA? And I think it's for a few reasons, but one of them is that you're amplifying things and it can be a little bit random what amplifies. And so the way to figure that out is with replication. So if you can sequence 3 replicates of at least a few samples, you can get a sense of what's random, what's noise in your data versus what signal that's coming from the world, coming from the ecological pattern that you are interested in. And this applies, I think, to all eDNA work. There's some of it is noise and some of it is signal, and we're trying to figure out which is which. And it's useful and important, I think, to interrogate your data in that way. Please, Stefania.
Yeah, I just wanted to make a comment. I was actually really impressed by also some of you guys tackling such huge methodological challenges like sequencing genome from the spout of whales or like viruses from the tracks of mammals. It's amazing, and I really hope it works.
So do we.
It's a great challenge, I think.
It's a challenge, but it's also like time consuming. Wanting to take on so many, like, I guess, side projects like that. Like, just for example, in my case, swabbing eDNA from footprint pads. I mean, at the basis, my goal would be to just have a pilot project and just see if it works. But then I have around 3,000 papers that that I'd have to swab, and it's just a matter of logistics, time-wise as well, and trying to find ways to make it most efficient is also a big challenge. But yeah.
I think it's a great way of learning the methods.
Yeah.
So it's not all lost in
Yeah,
a
of
way.
course.
So
Yeah, you know, it's funny, we talk about this a little bit at the collaborative here too, which is when we think about the field of environmental DNA, it's newish, but it's really reusing old concepts. And so it's going back and using the same tools. The tools are not new, the application is new. But I think sometimes we get stuck in a rut. Like if we're working with water samples, we say, okay, we need 1 liter and we need this extraction kit and we need this PCR protocol, but getting creative and getting out of that rut. So it is really fun to hear the sort of different applications and trying to not be stuck in our ways, because it feels like magic. Like you take a liter of water and you see 100 species, feels like magic. So part of us is a little afraid to change anything in the processing protocol because it seems amazing that it works, but not being afraid to try something new and pull from another field and say, well, if they do that over here, maybe we can try this. So it is really, really interesting to hear kind of the more creative ways of doing this. And I know diet, there's all kinds of interesting differences in diet versus, water samples and things like that. So hopefully, Stefania, you have some context there. We can put some more in the Google shared document too. We have a PhD student here, Mary Fisher, who did some crab diet work. It's a whole, everything has its own unique challenges, but hopefully reaching across and learning from other people will be helpful. Aiden, you want to go ahead?
Sure. Do we have time for more questions? Is that okay? Yeah. I was just curious people's experience on various filter types for seawater eDNA. I'm too far into this particular project, unfortunately, now to change it. But because we work so remotely, one of the big challenges here is trying to keep these filters frozen and then ship them frozen to get them back to the lab. And I know there are these self-preserving filters out there. They're just pretty expensive as far as I know. Yeah, people are nodding. So yeah, just curious if anyone has come across a self-preserving filtration method, or one where you just can avoid the hassle of freezing and then dry ice shipment. And that is a little bit more cost effective for small organizations. I'd be very curious.
I bet Eiley wants to answer that, and I will look up the link for Longmire's buffer.
I was going to see if anyone else chimed in first, and then we can be the sweeper crew, Ryan. But has anybody else have other suggestions?
Well,
Yeah, I was
also,
just thinking about, oh, sorry.
sorry, please go on.
No, yeah, this Canadian company that is the Waterra filters that are quite cheap and the long Meyer buffer conservation. But it's not my area of expertise. So.
If you have the link to that Canadian company or just the name of it again, because I'm in Canada right now, so that could be a potential avenue for us. Thanks.
I also read about like just dry preservation of the filter papers with what was it, silica gel in. So they put the filter paper in another paper box, and then there is the silica gel next to it in one zip bag, and you can store it.
Great.
But haven't used that so far. We use ethanol for preserving in small tubes.
Okay, yeah, I've heard of that. I also, oh, Diego has another suggestion, maybe.
Yeah, thank you. I mean, it is not directly related to this question. It is about actually the question on the bioinformatics as a bottleneck. So an important consequence that I was thinking of in the meantime is that actually bioinformatics in having a bioinformatician in ecology can be a bottleneck even for the bioinformatician point of view. So those that already are experienced and perhaps specialized in bioinformatics. With that, I mean that currently the labor market is way more biased towards positions in microbiology, like industrial microbiology, or medical biology. So actually, most of the expertise of bioinformaticians gets sucked in other branches of ecology because there is more interest and there is more money over there. And also, I have happened to find some position in which bioinformatics experience was required. And it is also common that in that case, not only bioinformatics experiences are prerequisite, but also skills in wet lab, which would make life even harder for a person with a primarily bioinformatics experience to be hired as a support in an ecology research group in an ecological context.
So I think to summarize that comment, and tell me, Diego, if I get this wrong, but that this is a struggle that I think we all face. We would like more bioinformatics experience and expertise. And sometimes we need to solve problems that we don't know how to solve. We don't have the coding skills ourselves, but bioinformaticians are expensive and it's difficult to find them. And usually someone else can pay them more. And if we want to hire someone, or if we are trying to work together, they need lab skills and coding skills. And that is a difficult combination to find in a collaborator, because those are different skill sets. If I have that right, I think that's just to sort of reinforce that. I think we are all facing that same challenge, and I don't think anyone has a solution to it at the moment. But my suggestion is that I think we. One of the ways that I think it's important to reduce barriers to entry and reduce fear is particularly around coding, around bioinformatics, because for many of us, we are interested in ecology, or we're interested in root microbiomes, or we're interested in forestry. We don't want to become coders. But I think that is, unfortunately, increasingly a necessary skill. And so getting over the mental hurdle of saying, okay, going from a place where you think, oh, coding is something that someone else does. I don't do that. I think we do that. We have to do that because I don't see another way forward. You want to be able to have the autonomy to own your data. You want to own your analyses. And I think the way forward there is to learn enough. You don't have to be a professional coder, but you have to learn enough statistical coding that you can write some code yourself and understand that. And I think that's very empowering. So I am a person who I had to come to that later than I would have thought. And I avoided it for a long time. And so I just encourage people to try, and if that frightens you, try and get over the fear of that. And there are good ways to do it. I think I've seen some things in the chat here with bioinformatics classes. The computing coder language R is quite useful in that it's free, it's open source, and it has a worldwide community in many languages of people using it. Same thing with Python. So I realize that's not easy, but I think it is increasingly necessary for our jobs.
I think we'll take a couple more questions, and then we have a few last slides and just reminders for people to really help facilitate more of the networking. So kind of like a last call for big group discussion questions. And of course, that Google Doc that Kate set up will be live. So if you have something that comes to mind later, feel free to add to it. But anything else before we switch back over? Okay, I think Kate is going to screen share again for our last reminders here. Get her back
Give me 1 second.
Maybe it's worth, the chat is going back and forth with what do you do with the Longmire's buffer? And are you looking for cells, or do you have things on a filter? And I think we all are going to have different, slightly different methods, right? If you're using soil, or if you're using stomach contents or things. So those are all different, but Longmire's buffer is a combination of preservative and lysis buffer. So it doesn't actually matter if you've got cells, if you've got a paper filter, if you've got soil. I think anything that a buffer can penetrate, right? Like you couldn't put, I think if you put a whole chunk of tissue into buffer, it wouldn't preserve very well. But otherwise, I think with cells, with paper filters, with air eDNA, with soil, as long as the buffer can penetrate, that I think will work just fine. I might have misunderstood that question, but hopefully not.
Do we have time for me to jump in on it? Is that okay?
Sure.
Yeah. It's just such a niche question, so I do apologize. I'm going to show you my issue. So I'm using Longmire's for our blow samples, not for the seawater eDNA. And we pre-purchased the bottle of it. And I don't know if you're able to see that there's a settled with chunks at the bottom that seem to not want to redissolve. So I'm like, before applying the Longmire's to our blow sample, so I use the Longmire's to wash out the Petri dishes, which has the blow in it. And I was just curious if that was an issue other people have, because if I'm quite vigorously mixing the Longmire's in order to redissolve the solid base at the bottom, it then starts to bubble up, and it's then an issue to get it into the cryoval. So it's just very niche issue. I'm just curious, people with experience.
That is a very specific issue. Yeah.
Yeah. Just when you mentioned Longmire's, I was like, here's my chance to ask someone who might know.
But I do think that is one thing that's common to everyone and to you. I think we have learned, and someone correct me if I'm wrong, but there is a lot of DNA in the world. So almost always you don't have to worry about missing the DNA, and as long you are getting when your buffer is coming in contact with that puck at the bottom or whatever, and you're agitating it, you're getting a lot of DNA. I think you probably aren't missing. You are losing DNA, yes, but you've got plenty probably.
It's just that the solid part is part of whatever are the ingredients in the Longmires. So this is the bottle of just the Longmires itself.
I misunderstood.
Sample is added.
I misunderstood.
It does precipitate. It does precipitate. And some of the Kyogen buffers precipitate as well. I think ATL, and I put this in either the chat or the Google Doc. I can't remember which at this point, but and I think heating might, rather than shaking, if you have any way to heat it, that might be able to get that precipitate back to dissolve, so that you can then it doesn't get all bubbly when you're trying to aliquot it out into your petri dishes or whatever. I would try heat. And then the other thing that I put in there is there's a couple other commercially made. I don't know if you make the Longmires yourself, but maybe trying another one like DNA and RNA Shield from Zymo, or trying I know people store in Kyogen ATL. So it might just be trying a slightly different one. The Zymo one, you can then, they have their own kit. So then you can just use that and go integrate really nicely into their kit for extraction. But it's also, I think, pretty compatible with, like, if you're using the Kyogen kit, you could use the Zymo preservative and then move it into the Kyogen kit. It's really similar. They won't exactly tell you because it's proprietary recipes, I think, but we've done some internal testing. But yeah, certainly. And if there's other questions, let us know if anybody else has experience with Longmires or their preservatives. And Stefania has a hand, so maybe she does.
No, I just wanted to comment that it's actually quite interesting. We are a growing community of users of eDNA in different ways, but we don't have a common platform when we can exchange these practical, like ask the questions and have this. So I don't know, this is perhaps something to think about. I am not exactly sure how. But yeah.
I'm just going to drop into the chat, which I should have put, and we can put this in the document, in the Google document too. There is, if anyone uses Slack, there is an environmental DNA Slack channel created by Peter Thielen at Johns Hopkins University. That link should work because I think anyone can invite anyone. And it's supposed to be open. And it's supposed to be, you can make your own channels. You can ask the community some questions. We use Twitter a lot, but Twitter has kind of been declining a bit. So people have different feelings on that, and whether really the strength of the community is then how many people use the tool. So we're trying to figure out if there is a better tool. But I will say that this Slack channel was created, I don't know how long ago, but please pass that around, because again, it's only as useful as the number of people who join it and participate in it. So please, send that, and I will make a reminder to myself to put it in the top of that Google Doc as well. But I completely agree with you.
Yeah. And also just a reminder, we do have the listserv, which is active and does work. I did check. So you are everybody who's on this discussion right now, and all other microgrant recipients and visiting scholars and other grantees are all part of that one listserv. So if you do have a question, feel free to send it. You should all have the ability to send things to that listserv address. I'll put that in as well so that you know how to do that. And then the microgrant community can at least get back to you. It's a smaller community, but it's also another option.
But no question is too small. If it's about buffer precipitating or anything really, we're trying to build these tools so that they're useful for all of you. And somebody else probably has, if not the same question, some experience with it. So yeah, please feel free to use those. And again, Kara mentioned it's a smaller community, but it's also kind of nice that it's smaller. And again, this is part of the meetup, is that you can put some faces to names and feel more comfortable asking any kind of questions from, preservative to what settings you're using in your bioinformatics and data 2 or whatever. So yeah. Okay, that brings us to our kind of wrap up slides here. And this is also to really try to facilitate, if anybody is going to any upcoming meetings, this is a screenshot of kind of what's coming up that's on our radar, that's conferences that people might be going to. And then on the Google Doc, please add, if you're going to something else that we don't know about, we will put it on our website. We will put it on our other Microgranty meetup meetings. And if you feel comfortable, we put a little place there for you to put your name. So thank you, Cleo. I saw you wrote that you're already at ICES here in person, but what we would love is for you to find another Micrograntee, find somebody else in our network and actually meet them face-to-face in person and have some of these conversations. So in the Google Doc, again, that's a live document. If you can add other meetings that we are not aware of yet, and if you would like to if you're planning on going to these meetings, putting your name. So I think the other one that's in there right now is in GeoBond coming up in October. Nuo, who is both a Micrograntee and a visiting scholar, will be there. He's very friendly. He'll be on another one of these meetings. So if you could email him and schedule a meetup with him. This is again meant to help all of you expand your networks and meet fellow people working on similar projects. So that's just on there. And then I think next up is going to be, we have tip of the day. So I think Kara is going to go first with networking.
It looks like you are already, most of you guys are already doing this, at least talking about it. But I think to get the network going, just pick somebody that you have enjoyed their talk, have things in common with, want to work with, and shoot them an email after this where Zoom is over, and that'll start to get things going. Also, before we skip to the science one, I'm just going to do a little plug for also, if you haven't already, feel free to write a blog for our website and send it to me at the eDNA Collab email address. And for those of you all that have done one already, thank you. You can see your posts online. That's it.
Good plug, Kara. And so then science tip of the day, that I think it's always really useful to be really clear about what you want to know from your data. And this is surprisingly hard. It sounds easy, but it's hard. And so if you sit down with a piece of paper and a pencil and you draw the figure you would like to be able to make at the end of your project, that will help a lot. You make decisions about what is the kind of data that would enable you to draw that figure. Like, do you want error bars on your dots? Like, if so, you need replicates. What is the figure? Is it a box plot? Is it a scatter plot? How many samples would you need to make that figure? And that really helps. As early in the process as possible, draw the figure you want. Draw your future, your preferred future.